Okay, so up until now we've been talking mono hybrid cross for simple dominance. And so remember, hybrid means a mix, in this case, of alleles. So we're talking heterozygotes, like big A, little a, cross with big A, little a. And this is exactly what Mendel did and discovered the 3-to-1 ratio kept coming up, and that's what led him to develop these ideas. But one of the incredible parts of this is that we don't just have to focus on one gene. We can do a dihybrid cross, or if we want to get crazy, a trihybrid, or a quad tetrahybrid, or whatever. Meaning we can focus on more than one gene at the same time. So we can add in a second gene here, and what we'll find is that the probabilities associated with the A gene can be looked at completely separately from the events with the B gene. Because if you'll remember, back to the events of meiosis, we've got these chromosomes lined up in meiosis, and they're going to split. And so when we're looking at the gametes that can be formed from either of these guys, so like we make our Punnett square here, big A, little a, big A, little a. The reason we're splitting up big A and little a is because that represents the fact that over here we had a big A and over here we had a little a. Okay, and so that gives us, our 3-to-1 ratio that should be easy for you now. Well, we can do the same thing with our B alleles, the same story, and it's for the most part assuming that the B's are on different chromosomes. It could be big B, little b, or let me erase that. It could just as easily be little b, big B. Okay, so, and it doesn't matter at all what has happened up here or down here. So each of these events is independent. And so that we'll use that to our advantage here. Okay, so it's going to look something like this. And what we can do is, if we want to know, I can ask you a question like, well, what's the probability of getting an individual that is AA, BB? Well, that seems like a complicated question, but really, again, we can separate it into two separate events. The chances of getting little a are 1 out of 4, so 1/4. And the chance of getting little b are 1/4. And if we want to know the probability of these two events combined, then we just need to multiply them. So 1/16. Okay, so this is kind of in some ways a shortcut, but it just takes advantage of the fact that we recognize these are two separate events. And it's a heck of a lot easier than drawing a 16 grid, A, B, A, B, A, B, A, B, A, B, and figuring it out that way. But you can see, hopefully, and this is kind of small here, but our AABB is going to come down here in this one grid out of 16. If you can follow my scribbles there. Okay, but so this is the 4 different possible gametes that we could make here. All right.